Now you know you're being recorded. Okay. So after this is over tomorrow, we will post this presentation. If you have to leave early or for some reason you get knocked off because of weather or whatever, we will post it on our Vimeo channel, and the link to it will be on the Bear Audubon website under our programs in case you missed it. So with that, I'm going to be quiet, turn off my camera, and turn it over to Dr. Athrey.
Thank you very much. Thanks for the great intro and for the invitation, Tom and Patsy. So it's a pleasure to speak to you all. So I want to start by actually kind of briefly talking about how important it is for people like me who are doing these kinds of work to speak directly to the general public, birders, people who are obviously not, this is not what they do for a living. And so Patsy mentioned that we met in 2015. I gave a talk, and it just so happened that other birding organizations thought it was worthwhile to hear what I had to say and talk about our completed studies at that point. And so I got invited to talk at the Texas Ornithological Society, and then Bayer, Travis Audubon Society, and a couple of other places. And it's just that you reach more people and the advocacy becomes, interesting, or enough people hear it that they think they need to do something about it. And one thing led to another. And eventually, when the U.S. Fish and Wildlife was interested in performing this type of study again, they knew that I had been talking about it, that I was interested in doing this again. So in a way, what started on that day at Guadalupe River State Park is, directly responsible for what's happening right now with this study. And hopefully that's going to, again, this moment will carry on, and it'll keep leading to further discoveries which are useful for conservation. So thank you very much. I can't overstate how important these types of things are. So as Patsy mentioned, I'll be talking about the genetics of the Golden-cheeked Warbler. And most of you have seen these birds in their natural habitat, these beautiful, what I like to call, sentinels of Texas heritage. And, we'll be much poorer without them. And I want to acknowledge my two collaborators who have been very key to this work this time. So Dr. Laurel Moulton, who is a biologist at the City of Austin, and Lisa O'Donnell, who is also a biologist at the City of Austin. So they have been, central, crucial, critical. You choose your particular word for that, but they have been very, very important for conducting this work. So a million thanks to them. So let's talk about what exactly we are doing and what that means for the future of this bird. So as you are aware, this is obviously our the bird of interest. That's actually a picture of a Golden-cheeked Warbler in my hand, taken in 2006 when I was doing field work on them. So you probably see a silhouette of a person in the background. So that's me trying to take a selfie with this bird. We know that the breeding range in Texas is obviously shrunk quite a bit in the last century, and certainly continues that way to this day. And the winter range is also under pressure. So because everywhere we in not just Golden-cheeked Warblers, but it's a representation of the natural world nowadays that human activity is just putting a lot of pressure on lots of wild species. So in a way, this is an argument for that many conservation biologists use, in that we need to protect endangered species because they are a representative of what's happening across that environment. And many species are similarly affected. Perhaps they aren't showing the worst of those effects yet, but that's the point of conservation, that you create this umbrella of protection and it helps many other species. And as we know, one of the things that decides these types of protections is how abundant these types of animals are. So golden-cheeked warblers were listed in the early 90s, and at one point they were believed to have been, a very low number of pairs. I believe 100 and odd pairs of birds left when it was listed on the endangered species list. And through a lot of management action and protection, now we know that they have somewhat recovered. So there appear to be a lot more birds nowadays than they were counted as. Now, one of the problems with these types of situations is it's very hard to know whether a species has been historically rare or it was highly abundant. So that's what this particular chart is trying to kind of give you with these question marks. Was it something that was always abundant? I'm sorry, always rare, or was it something that was very, very common? And then, mostly during the time of human activities or increased human activities, they have become, progressively rare. And so that's a question that is difficult to answer, but this is what we tried to do in one of my previous studies, so which I will give you a brief overview of. But more recently, we know that there have been a lot of work on the habitat side of it. So genetic studies are one way of looking at these, because, and as I'll talk to you, we can learn some things about what's happening in these species, but other kinds of studies are also important. And one of them, a common one, a low-tech way, if you will, is to just go and see how much suitable habitat is there, try to count how many birds there are, things of that nature. And some of these modeling studies, where they use, say, tree cover from satellite data, or use a particular habitat and then try to extrapolate it to other regions, have come up with some different kinds of estimates. So, for example, they think that habitat is now much larger than it used to be in 1990. And similarly, if you see on the left-hand side, the population is also supposed to be much bigger. And this is one of the reasons there's increased, I guess, some, views that they deserve to be delisted, just like the black-capped vireo was delisted a few years ago. And so, of course, this is, a little sort of a cognitive dissonance, because if you think about it, there's actually this period that we are talking about has seen a 30% reduction in warbler breeding habitat between 2001 and 2011, based on some other land cover models, that is, that data that's available out there. And secondly, also, just looking around and driving through Central Texas, no one will be confused that now you have more wilder regions, more forests than there used to be 20, 30, or 50 years ago. So there's something that's missing in these types of pictures, which say, okay, well, there's a lot of birds, there's more habitat, so therefore they must be delisted. So these types of, debates are very difficult to settle, and reason these kind of stick around for quite some time. So now that's where some types of other approaches are required as a tiebreaker, if you will. So because genetics is something that is, not so much of a subjective approach, it's more of an objective approach to understand what's happening, at a level where you're not counting the birds, but you certainly see the effects of the actual count of the birds. So because, one of the things we are interested in is that any population, if it has to survive for a long time and succeed, it must have abundance of genetic diversity. So I will talk a bit more about genetic diversity, but that's hopefully a concept that is generally making sense to most of you. More the genetic diversity, the better prepared organisms are to face challenges, including things like diseases or climate change. So for example, you might know that just this year from the bird flu, some migratory bird populations have crashed, significantly. So especially in, like, I just looking at a news article in how in Michigan, tern populations are really under pressure because of how many mortalities there are due to avian flu, which typically is over by this time in the summer, but not this year. So genetic diversity is considered key to being able to overcome these challenges. It doesn't mean all the birds are going to survive, but genetic diversity is important so that some of them may survive and pass on those advantageous genes to the next generation. Same thing goes for people who survived the 1918, influenza pandemic, or people who are, who say maybe didn't get affected so bad by the coronavirus pandemic. So it applies the same way to us as to any other organism. So these are the types of things which can be a different or alternative view of what's happening in a population. And so the previous genetic studies that we conducted showed that there was a significant decline in genetic diversity over the past century. And we based that using historical context, because you cannot measure something at one point in time and say, well, we know what was happening in history. So, but if you have 2 points, you already have a lot more information. And so this is the approach we took in this previous study, which was published a decade ago, where we measured different types of or different metrics of genetic diversity in golden-cheeked warblers from those that were collected in the late 1800s and early 1900s. How did we get this data? I went to museums all over the country and went to their collections and took toe pad specimens from them, from which we could isolate DNA. And then we compared them to samples that we collected from contemporary population. So this was in the between 2006 and 2008 that I collected myself. And so by comparing what was happening over this 100, 120 year period, we were able to generate some sort of a historical context for the genetic diversity we see in populations right now. So that's what this chart shows. So you see time on the x-axis. I'm sorry, my mouse is not responding to this, but hopefully you can see the time on this axis. And there's 2 types of genetic diversity measures that are commonly used in these kinds of studies. One of them is called heterozygosity, which is on the bottom, and allelic richness, which is on the top. And we looked at these where we were able to compare 3 different regions in Texas. So Kerr is a place where we had good representation from historical and contemporary times, similarly for the Bexar County area and also Travis County area. And so that's what these lines, each of them represent. And these were, we compared these measures of genetic diversity using statistical approaches, and they showed a 20 to 30% decline on average, which is pretty significant in just 100 years. So that roughly corresponds to about 70 generations for 70 to 75 generations for these warblers. So that's a long, that's a lot of decline in genetic diversity in a pretty short time. Now, heterozygosity is something that actually changes a little bit slowly compared to allelic richness. And we see that even though that is the case, it was also showing a great amount of decline. So this was definitely concerning for the warblers. So until now, this has been the last word on this matter about how much genetic diversity was lost in the species. And obviously, anything we are going to look at, we'll have to consider, well, what's happening in the future and how does it add to this picture that we've looked at? How does it relate to anything that we have seen so far? So these types of declines in genetic diversity are something we see with significant population declines. So here's an illustration of what happens where each of these colored balls are kind of a representation for genetic diversity. So the more number of colors you have, the greater the genetic diversity. And in a typical population in a normal or unmolested wild population, you might expect to find a lot of genetic diversity, but there is a sudden mortality event which can cause the removal of the majority of that. And so if you can think of the tern mortality I talked about because of avian flu, this is at least on a local scale, something that would be happening there, because only a few of those quote unquote genotypes, which represent the specific types of diversity in that population, are surviving. And so all future populations then will be represented by these limited types of varieties in that population. So when this happens repeatedly, or when the kind of a bottleneck is very dramatic, when basically you're left with a very few types of alleles or varieties of genes in these populations, then you have a bigger problem, because that limits their ability to respond to these challenges in the future. So if you again go with the tern example I've been giving, if they were to come back next year and the same thing happened, the selection would be even stronger. So perhaps only a very small number of those could survive that second infection. And so that might theoretically reduce their genetic diversity for that population even further. So how do we relate this to golden-cheeked warblers? So the pattern we are seeing here basically suggests that this type of a bottleneck has occurred or continued to occur over quite some period of time to observe the amount of decline in genetic diversity that was seen in these populations. Now, the other thing we looked at was how much of these individuals from these populations, so Bexar, Travis, and Kerr counties, how much were they exchanging or mating with each other over time? And this is something that could be estimated by another genetic analysis, for population structure or gene flow, using a parameter called FST. So this parameter can be a little counterintuitive. So the greater the value of FST, that means lesser the amount of gene flow. So what we can see here is in the historical period, which is the salmon color over here, the FST values were low, which means there was a lot of gene flow. So 100 years ago, there was much more mixing of these populations. Whereas, 100 years later, there's much less movement of these individuals across these sites. That means they aren't moving that much. So all the genetic variation now is kind of sequestered, segregated within these units, which poses a problem. So if one population experiences some dramatic event, then you're probably going to lose some very unique variation in that population. So ultimately, the genetics help us understand what are the threats to these kinds of species over time, but also across space. So what should we be careful about so that it can be managed not just over time, but also across space? So in this current project, these are the types of things we are trying to revisit and provide more context on. and help the fish and wildlife, include the latest information so that they can decide how to deal with the management and any potential delisting questions. So the first one was we came up with in this study is to completely modernize the way we are able to understand the genetics of the species now, but also for all eternity. And I don't say that word lightly, because that's what I hope we will be doing, because unlike the previous studies that we did, at that time the genetic technologies were dramatically different. And in the last decade, the field of genetics has seen some significant improvements, because you probably know in 2001, the human genome was sequenced. And I'd be surprised if one of you among this audience is not also has had your ancestry figured out by ancestry.com or 23andMe. And that's basically an extension of that technology, which has gone from a $3 billion project, which was the Human Genome Project, to a $30 ancestry test that you are able to purchase online now. And the same types of revolution in these genetic technologies was just not available for us when we did this back then, but now they are. And by adopting these approaches, by building a completely new framework for how we can answer these types of questions, we decided we're going to set the foundation for how these questions can be answered forever. So there's no questions about, oh, that was a different method, or this method is better than that. No more of that, because this is going to be the gold standard for how these studies are done. And this is, it's not unique to our study. It's pretty much how all animal studies and human studies are approached these days. And it is to build something called the genome reference. And so that was the first thing we set out to do in this project, to generate a so-called de novo or from scratch genome reference for the Golden-cheeked Warbler. And this will be made publicly available for anybody to use. And if anyone chose to perform another study in the future, this will be a resource they can use to understand how exactly genetic diversity or any other questions related to patterns of genetic variation in the species are changing across space or time. So that's the number one thing we set out to do. So what exactly is a genome reference? So I want you to think of anything you're going back to the last time you had a genetics course. I don't know, it may have been some time for some of you. So you might know that we have chromosomes. No, Patsy will give you a prize if anyone can tell me how many chromosome sets you have. Let me see if anybody's trying to answer this. No. Oh, there are questions for me. There are 46 total chromosomes, 23 pairs. That's right. Yes, the habitat. Sorry, I was not paying attention to these things, but let me quickly go through this checkpoint. Some very good questions. I'm sorry, I neglected these. Population change real or a function of better counting techniques? So counting techniques have changed a little if you are strictly asking about counting techniques, because it's people use transect methods where they're walking out in the field and they're trying to listen to the sounds, and then they feed that data. Things that have changed are imaging, satellite imagery for habitat cover, but all this data goes into a model to kind of come up with an estimate. So partly some improvement in methods, but as far as actual counting methods of birds, not a whole lot of change there. Now, I should say that there are some recent efforts to apply some GPS tags on birds so they can track them, especially during migration. But those things are still, they're not so much for counting, they're just trying to understand the biology. Next question is, I'm not going to comment on that, about the comptroller's intentions. I am a state employee. Habitat map for 2022. Yeah, so this is this map will look different based on who is going to provide you one. So but yes, I can actually share a more some examples of recent maps. I'll share it with Patsy, and I'm sure she'll be happy to share it with you all. How do you sample the genetics on living birds today? Yeah, same approach as we've always done. And great question, thank you for this. By misnetting, so targeted misnetting, playback of the song, and catch the males and the females if they do come in, and a small pinprick on the brachial vein, and to collect 10 to 20 microliters of whole blood, and that's our source of DNA. Will we go back and analyze the samples you used in the first study using new genetic technology? Well, from the 2006 studies, yes, that is the intention. We still have some of those samples, and we plan to include them, not immediately, but yes, we are working on that. As far as the museum specimens, they are valuable resources. That's what was sampled for the first time. We don't have anything left, but the birds are there in the museums, and it's, if we can provide sufficient justification for why we want to do this, they probably will be inclined to share it with us. And we don't see a problem why we can't do this, because Neanderthals who have been dead in caves in Siberia for 13,000 years have had their genome sequenced recently. So I'm pretty sure we can do it for golden-cheeked warblers from 100 years ago.
All right, I don't want to, Giri, I don't want to interrupt you, but on the screen, we're getting a black box in the middle of your slide. I don't know.
That's where the chat, that's where the chat window is.
Oh, you have the chat up. Okay.
Because I'm looking at the questions. I'm looking at the questions.
Okay, very good. So it'll go away as soon as.
It'll go away. It'll go away.
Okay. People want to see every.
Yeah, I think it blocks it. So I guess want to keep the chat messages private, but I will remove it. Does anything take place during the winter migration? Ameliorate any of the lessening of genetic? So this is not particularly winter migration itself. On the wintering ground, sure. I mean, there are the same pressures in Guatemala and some other regions. I'm not the best person to answer this question. There are some very good people working on this, south of our border on these questions. Habitat loss is a concern there also, broadly speaking. But other than that, I think most of the issue could be breeding habitat, shrinkage of or at least making a bigger impact. Yes, okay. All right, so I will get back to my talk and we will revisit these questions. If I can make this go away. I'm sorry.
Yeah, we're still seeing it.
Yeah.
Yeah.
Oh,
just reshare your slides and maybe it'll go.
Yep. Okay.
Some great questions. Thank you, guys.
Absolutely. Okay, back to this. So that's how we sampled the blood, as I described. The genome reference for the golden-cheek came from a bird that we sampled on the Balcones Canyonlands Preserve in May of 2019. I'm sorry, May of 2020, just before everything shut down. So, so what is a reference genome? So quick overview of this idea. Reference genome is a representation of a species genome. So you all know that we have chromosomes, or our genetic material or DNA is organized into chapters. We can call chromosomes, and similarly for other species, birds have different numbers of chromosomes. In the case of golden-cheeked warblers, probably between 35 and 41 pairs. We don't know the exact number. We won't know based on the methods we're doing, because those are something we get from other techniques called cytogenetics. But a genome reference tries to sequence or read every single letter in the alphabet of the genetic material, so the DNA, and put them together. So basically build out this manual and understand what's there, where is it located, and where possible, what exactly does it mean. So this reference genome can be constructed from a single individual, very common now. Previously, you needed a lot of DNA because of technical problems, and you had to use a pool of individuals. And then the genome references can provide some information about the variation in a population. So these are called alleles, so alternate forms of any gene present in an individual. But this is not the purpose of a reference genome. It is just a reference for what a genome looks like in the species. It doesn't do anything else beyond that. What it's useful for is comparing multiple individuals of that species. So now that you have an idea of what this manual looks like, how it is organized, what's the content of this genome, we can compare the same pages and the same passages of this manual across multiple individuals and see what's different about them, whether they all have identical passages. If there's a lot of identical passages, that means less genetic diversity. If there's a lot of unique passages, that means high genetic diversity. That's how we assess these things. And genome references at best can be organized into chromosomes or so-called scaffolds, which are not the best, but these are technological limitations on being able to do these. And worst cases, these so-called contigs or super contigs, which are acceptable, but it's not the best. And it's not that you're stuck with one and you can't do anything. These types of projects proceed in a stepwise fashion, starting at the contig or super contig level, and then you improve them by adding additional data or applying new computational methods to make it into scaffolds, and then continue refining them until you have a full picture of what these chromosomes look like. So the challenge of assembling a genome is like this, but actually, in reality, it's probably worse. So imagine that a sample of blood from a bird has millions of copies of the same genome. It has the same genetic material. So because every cell has, a copy of its genome, the number of chromosomes they have. And if you were to sample, thousands of cells, they all have these thousands of copies of the genome. So you can think of these as newspapers, but in the process of trying to build the genome assembly, you're going to have to isolate the DNA. And when you do that, you end up sharing them, and it breaks up into these pieces, which is also required for this technique called DNA sequencing. So you are left with this shredded pile. And then your task is to put it back together into a finished product, which is actually useful. So I'm using a newspaper here as an example here. And so that's the perfect scenario. It doesn't always turn out so nice because, as I told you, there's contigs, there's scaffold, and the chromosome level assembly. So this very nice looking newspaper copy would be your chromosome assembly. Typically, this is how the process goes. So you have an actual genome, which is unknown to you at the beginning, and based on the methods or how good or bad the whole thing was planned. It could perhaps be unknowable. But you start with your DNA, which are represented in these so-called sequenced reads over here. That's when you use the DNA and put it on a sequencer, and you generate short sequences, which are read from the sample that you used. And then you have to put it all together in the correct order. So this is actually, quite a challenge, if you can imagine. And the typical assembly is going to look like this, represented by the blue bars. And you have some pieces missing from it, which are still hard to put together. So actually, in the year 2022, a couple of months ago, if I'm remembering correctly, the human genome released something called the T2T assembly. So a full 22 years after the first genome assembly for humans, they generated this so-called telomere to telomere assembly. It took that long for them to actually build out a complete genome reference. So obviously you can think of other species are not exactly there yet. So it's going to take a lot more work. But it's a process. The technologies are better. It won't take us that long, but it's still that process. And so what did we do? So we use this approach, and we use different computational methods to assemble this genome, starting with this pile of shredded DNA. And you can see that we're trying to understand how our genome assembly is improving through our repeated processes that we're working on. And don't worry about these terminologies and the methods that are on the top of this table. What I want you to kind of only pay attention to is this thing called N contigs, number of contigs. That means the number of large pieces that we are fairly confident of represents actual pieces coming from the warbler DNA. So our initial attempt provided 4,900 4,900 and odd sequences, which were pretty big, but it's a lot of pieces, very fragmented. But through repeated processes, we have got into an assembly which now has half the number of pieces, less than half the number of those pieces, and also the longest contig, which is the second row over here, will show you how much better the continuity of these pieces are. So it went from 4.9 million letters, so million base pairs, to 12.6 million base pairs. So we've improved this assembly quite a bit. And already this assembly is better than 95% of all vertebrate assemblies that are out there. So it's highly usable. Doesn't mean, even though this is fragmented, it can't be used. We had to wait for another 5 years. No, it can be used right away. How do we know? Well, we can assess something called assembly completeness with this. So this allows us to see, based on everything we know about bird genomes across all species, everything from chickens to peacocks to bald eagles, how many genes are we recovering which are complete? So this is a particular analysis which benchmarks the quality of the assembly we generated. So what you want is this blue area, which represents the complete pieces, complete and single copy genes that we were able to recover, and it's about 65% in this assembly. So it's actually 70%. So this is pretty good for an assembly which is so new and for one of our first attempts. So this is obviously something that will be improved in the future, but it's already highly usable. So that means 60% of the known genes that you can expect to find in any bird species, we have put them together correctly. That means we are ready to go in and start comparing what those genes look like across individuals in the populations across Texas, and see how do they compare. Are they all having sufficient diversity? Are there specific populations which are showing reduced diversity for specific types of genes? For example, genes that may be involved in immune function, or genes that may be involved in surviving heat stress because it's so hot, or other things, or metabolic performance because they need a lot of oxidative capacity to migrate and fly for thousands of miles. So we can look at specific genes and compare them across individuals in Texas and say, what's happening here? This is almost like what people do in human studies. You look at large populations and we try to understand how are these two types of populations different. For example, what's different between people who have diabetes versus those that don't, or people living in Iceland versus those that are living in Florida, for example, just as an example. These are the types of questions that you can ask with this kind of data, and we are actually ready to do that. So that brings us to the objective 2 of this project, and which is to perform a genetic assessment of golden-cheeked warbler population genetic diversity and structure and population size history. So without this genome reference, we cannot answer some of these questions as we'd like to. And these are very crucial questions as far as the Fish and Wildlife Status Review is concerned. The main reason we want to do this is to update the work that I already talked about, and another older work by Lindsay et al., who's my friend and colleague from back in Louisiana, and who also talked about some of these same issues. So how did we do this? So we actually sampled at numerous sites across the breeding range. So 13 different sites across Texas. And as someone was asking a question before, how do we sample these birds? This is how we do it. Capture the birds using song playback. The person in the photos are Laurel Moulton, who was our collaborator from the city of Austin. She did most of these sample collections. I went out only once with her, so this entire project is possible thanks to Laurel. And basically, in every place we managed to collect, or we targeted between 15 and 25 birds when possible, because we think that's the number where we can actually have a lot of trust in it being accurately representative of what's going on in these populations. It's not too small of a number. It's sufficient enough of the breeding individuals in the population in each of these locations, so we know exactly what's going on between them. So questions that Fish and Wildlife are interested is, are birds between these locations showing any strange patterns of genetic diversity? Are they moving between them as we might expect? If the habitat was great, if there was no problems, are there any barriers to these gene flow? And what can we also talk about recent histories of their populations? Something that we can estimate by using some methods called population size estimations or effective population size estimations. I'm not going to be talking about that today, but various approaches. So here's what we ended up sampling. This work was done in 2020 and 2021. So 2019, 2020, and 2021. So we sampled over 3 years and captured across all these places, all these counties that you can see here, a total of 282 samples. So this will be the most expensive and most comprehensive sampling for a genetic study in the species when we are finished with it. And we have all now sequenced the DNA from all of them. Well, we have sent them off for sequencing. We are waiting on the results from all of them. That's one of the last things that we need to start answering these questions. And so these use some techniques called next generation sequencing. That's what we use. So again, a very standard approach, which is going to be highly repeatable for anybody who wants to do this in the future, including ourselves. And we aimed for a very high coverage of each genome. What does that mean? Well, the genome size of a warbler contains roughly about 1 billion letters, 1 billion base pairs. And because what we want to answer with these kinds of genetic data is very dependent on the quality of that information we generate for every individual, we need to rule out any possibilities that there's mistakes in the data, false positives and false negatives. And one way to do that is to have a high coverage of the data. That means, in effect, we are actually sequencing every individual 15 times. So that's a huge amount of effort, so that there's no technical reasons which may cast doubt on what we're going to find here and say, oh, we didn't have sufficient data, or you're not clear about these particular regions, none of those things. So that's more than sufficient data. And again, this is way more than what most conservation genetic studies are using nowadays. So we set a very high standard for what kind of evidence we needed, because if you're going to make any claims one way or the other, it must be backed with great amount of evidence. So we don't, as I mentioned, we don't have a whole lot of results to present at this point, because we are still waiting on some of this data to come back from the genetic or genome sequencing facilities. But we have some things that we've been able to complete based on the genome reference that we've assembled. So one of them is the so-called genome-wide nucleotide diversity, which kind of represents how diverse is a genome on average, just by looking at a base pair level or alphabet level data from any genomes. And this is a common approach for these kinds of data sets now. And what we can see is for in each of these colors and the dots on them, they represent individual places where we have sampled across this genome. So each of them are actually showing a different segment, a different 50,000 base pair segment across the genome. And each of these bars here, so there's the pink and the blue and so on bars, they are a different scaffold. So the biggest one is obviously 12 million base pairs, and then the others are subsequently smaller in size. And so what we can see is there's some regions which have very high diversity and some which have lower diversity, and this is perfectly normal for how these kinds of data show up. So we always look at the average nucleotide diversity. And for golden-cheeked warblers, this number currently is 0.0014. So this might seem like an abstract number, because it's like, what exactly is nucleotide diversity of such a small number? But because the genome is so big, it tends to be that this number tends to be kind of small. Now, how do we can always compare this to other species, some of them which are more abundant or rare, to understand where exactly this falls. So I have some representative here. So you have the Physidula flycatchers from Europe, which are much more than this number, 3 times more nearly than golden-cheeked warblers. And then hooded crows, which are a little less than golden-cheeked warblers. And then we have wild jungle fowl from Asia, which are more than golden-cheeked warblers also. So we see that it's within the range of what we see in other wild bird species. Just this alone isn't telling us whether golden-cheeked warblers have less diversity or more diversity. We can only answer that question once we have data for all our 200 and odd samples. So that's going to be my next update. Whenever we have that data, I'll certainly be happy to update you all. There's one other type of analysis we were able to perform, which is very useful and informative. This is trying to understand historical demography of the species. What this means is use the whole genome reference to reconstruct the past history of the species. Now, I won't go into the nitty-gritty of how this method works, but this is another well-established method. that has been used across various species, including humans, to understand how abundant or how rare was a species in the past. So if you look at this plot, on the x-axis, it's representing time, but you'll see that they're actually on a log scale. So on the extreme left, it ends at 10,000 years. And the next one down is 100,000 years, 1,000,000 years, and so on. So it doesn't tell us anything about the recent demographic history, this particular method. All it brings us to is, about 10,000 years ago. And what we can see is this species, or ancestors of the species, were, they were less abundant based on the y-axis here, which is the effective population size, so which is in 10,000. So roughly in the range of 20,000 breeding individuals in, between 100,000 and 1,000,000 years ago. And then during the last 100,000 years, this species had expanded its range and kept getting progressively more abundant until 10,000 years ago, when it was perhaps near its most abundant, roughly 80,000 breeding individuals. So, and that's the cliffhanger, because we don't know what's happened since then. And again, that's another kind of data that we can only answer once we have all our other individuals sequenced using other approaches, which we haven't been able to work on yet. So that's a bit of a cliffhanger, I know, but at least we know we've answered one important question, that historically golden-cheeked warblers were much more abundant, leading into the, towards the end of the last ice age. So, think of the time when the lost maples were lost, basically. And around that time, these birds would have been very abundant. And then since then, we know that currently they are much less abundant. And exactly how and when they started to become less abundant, that's the part we will try to answer with the rest of the data collection in our project. So as I mentioned, these are some other things that we are waiting on. So this just talks about some of these things that are in progress, hopefully. And if you are so kind enough to invite me again, once we've completed this, I shall be happy to share this information with you. And with that, I'd like to thank the numerous people who've been instrumental for this work, our funders for the US Department of Defense, the people who helped us with this project in various ways, and also, instrumental here. I should mention, sorry, I guess I somehow forgot to mention Tom and Patsy Inglet, who also belong in this list. As I already told you, this all started with just presenting and showing an interest in talking about this issue and having more people understand what exactly is going on in the species. So I thank you very much again, and I'm happy to answer any questions you have.
Okay, thank you very much. All right. Well, there's plenty to come. It sounds like you're just on the verge of an explosion of new information. We do have a few other questions that came up in the chat. There was a question about how many chromosomes does the golden-cheeked warbler have? Do you know?
Yeah. I don't know for golden-cheeked warblers because it would require us to do, as I mentioned briefly, another method called cytogenetics, chromosome painting techniques, which are, or karyotyping, which is required to count the chromosomes. So you might, if you think chromosomes, you have these little X-shaped things on a slide. That's how they count them. To do that, it's a different kind of approach, and we weren't able to do that for various reasons. Anyway, based on other songbirds, we know that they have anywhere between 35 and 41 or 43 pairs.
Okay. Well, that was the other question. Do all bird species have the same number? And of course they don't.
They don't.
And I think it's important for people to realize too that the number of chromosomes is not as important as what's on the chromosome, because like crawfish have 210, and we have 46. So I mean, are you smarter than a crawfish, you know? But it's not the number, but it would be interesting at some point to find out how many chromosomes they have. So is the idea to go all the way to finding out what's on each chromosome, like the human genome has? Is that part of your scope?
Not in this current ongoing project, but that's, I mean, because we are kind of the what shall I say, the stewards of this approach. At some point, it is in our purview to do that for sure. So it requires some, additional techniques and different approaches than we've done so far, because those are improving the genome to the extent of, like I said, the telomere to telomere assembly is a very intensive and very, very expensive process at the moment. And so that was not on the cards for this effort. But yeah, when either the technology becomes cheaper or there's a newer technology, yes, at some point.
And the way things are going, it's going in that direction. There's a lot of progress in that. Okay, well, is there anyone else in the audience who has questions in their mind right now and you'd like to ask? You can certainly unmute. And somebody wants to know, is that a salvia print on the wall behind you?
I'll let you guess. I will let you guess what you think it is.
Let's see. It's a bird. It's hard to see. I see the one that is the Carolina parakeet. What is that? Is that a painted bunting?
Yep.
Who was that? Who painted it?
You need to guess. Your namesake.
Really? Audubon. Well, I know he painted the Carolina parakeet one, but the other one too. Okay, so there's birds. So that's the answer to the question, and whatever plant that the bird was on. Yeah.
Yes. Yeah.
Okay.
And I have another bird painting over there. So.
Well, I have a question about the range calculation that you had at the very beginning of the presentation. You had said that the range of the Golden-cheeked Warbler had increased, and I can't remember the exact numbers, but the order of magnitude seemed to be about by 3x. And I'm curious to know more about why that range increased and where that range increased to. That seems pretty significant.
Yeah, great question. So the source for that data is actually the Texas Public Policy Foundation's Center for the American Future. So that is the source of that statistic. And the data was generated by some studies performed by other researchers at Texas A&M, if I'm not mistaken. So it was based on modeling studies. So they use point counts and other survey methods, not just point counts, I think, if I remember correctly. And then they use habitat suitability models. So it's a lot of GIS approaches. And based on that, it is an extrapolation where they say, well, if this habitat or this patch, which we've studied intensely, looks like this and supports this many birds, how much habitat is there and how many birds are there? So it's, that's the kind of approach they take with these studies. And that's the source of that information.
What do you think that habitat fragmentation has to do with the problem that you're solving or trying to solve, or the question you're trying to answer?
It has a lot. So it has everything to do with that, because one of the primary things is, if let's suppose fragmentation has increased in the last decade or 2 decades, then what exactly does that represent for habitat? So in many species, including our own 2012 species, show that when they are separated by major roadways, or there's unsuitable habitat in between particular population sites, then you have less gene flow between them. So there's a lot less exchange of alleles between them and then the smaller some of these units are, they are more at risk from random events. Also, they could get lost if there's some weird thing happening in a particular location. And also, the less mixing there is, then you start to see different phenotypic or what I should say is organism level outcomes. So recently, there's also been people studying changes in the song. So song usage, variation in the songs in between different sites, for example, between Fort Hood and even Bexar County, if I'm not mistaken. And there's in other species, they have shown that these are a part of the fragmentation because they're not hearing the full repertoire of potential songs in that species. Songs and song learning is a strongly genetic trait. So if you're losing variation for those traits, that's going to affect them as well. Then there's this, at this moment, I think anecdotal data about how the pandemic affected singing behaviors in some birds. So for example, when everything went quiet, birds changed some of the frequencies they were singing in, and they actually added new songs. And then when the noises came back, they had to change some of these behaviors again because some frequencies just don't make it across some distances. So the pandemic just proves the point that these kinds of things are happening in highly urbanized or disturbed areas. So it's not anything to do with the pandemic, but that these things are happening in bird population. So yes, so fragmentation has everything to do with what happens at the bird level, and many of these traits are driven by their genetics, and their ability to change and adapt is driven by their genetics. So if there are potential alleles associated with their how they learn their songs, then that could be affected as well in the future. So there is also such a thing. We tried to actually study this, but we were not funded for this project. But we were going to look at a particular kind of temporary deafness that occurs in extremely loud environments, and this has been shown in some birds, and it can have some other implications for their normal mating behaviors, for example. So
we have two
sorry,
more
that
questions.
was a very long answer, but hopefully I did answer your question.
That was a very good answer. Thank you. There's two questions that we'll end the night with. Is there any international research in collaboration with non-breeding areas like in Mexico, Nicaragua, where they're not breeding?
Yes, there is. I don't collaborate with them, unfortunately, but the Golden-cheeked Coordination Group is a group in which I'm kind of part of, and last time they did have researchers from Mexico and Guatemala, and the GIS or GPS study, I believe, also has some collaboration with them. Hopefully, I'm not remembering it wrong, but yes, there are some works going on with those groups as well.
And the question that would be important to all of us here is, how do we help keep this research going? I think you realize that we do Golden-cheeked Warbler surveys at Friedrich Park and in and in Eisenhower Park, other areas. Bear Audubon has been working on this for 15 years or so and publishing the information in reports and so forth. What else can we do as citizens of San Antonio and Bear Audubon people and bird lovers and people who want to keep this species diverse and strong? What can we do to help?
So, well, one is if you're already, adding data to eBird, and so I think, definitely continue that. I think it's a great, resource that adds to this information. But also, and I can share this information with you, there have been some concerns recently about how to use eBird reports and what they actually mean, and which might be useful going forward in perhaps changing some practices. I'm not saying what you're doing is wrong, but just being aware of some of the concerns that have been raised with eBird data and how relevant it is for some questions. I think that may be one thing. So I will share that with you. The other is, advocacy, of course. So public education. So you all are doing a great job recruiting new birders from everywhere, get them interested in birding and the importance of this. But what I want to, and I'm volunteering for this, is if you are interested in, developing brochures or pamphlets focused on say Golden-cheeked Warbler conservation, I'm happy to contribute material to it, help with the designing of it, help with the messaging. And I'm sure there are lots of people who work on these who are happy to help with this also. But I think that could be very valuable, because who are the people who are likely to care about these things? They are going to be who can actually do things about it and get bothered enough. They come from every walk of life. I couldn't say they're only one type of person, but I think generally though, and especially what we try to do at universities is try to make young people aware of these types of issues, so they can understand the scope of the problem and what they could potentially do. So anytime you want to do an outreach event, part of Audubon, Bear Audubon, you want a speaker, feel free to call me. If you want to develop materials, feel free to call me. This is important stuff, and it's part of what and why we're doing this also. So,
well, we thank you, and somebody just put it in the chat very well. We want to thank you for this wonderful work that you're doing, and I know it started off kind of slow with you, and then all of a sudden you started getting more and more into it, and people said, oh, well, here's a guy who wants to know more about that. And it's just kind of taken over, I think. But it is so important. It is such an, here's another one. It says an art contest among school districts highlighting warbler conservation. We have acorn clubs in the elementary schools. We'll make them available, things like that. Coloring books, all kinds of things of every level is very important. So anybody has any ideas, please send them to Bear Audubon, and anything that we can do to implement them or help you implement them, we will be happy to work with you. And so thank you so much for your time. I know it's a busy time for you. You just started school again, and we're really pleased to have you. And when you do get your other results in and you're ready to give us another report, we'll certainly love to hear about it and hear what you're learning from it. And I also want to thank one of the questioners here was Britt Coleman. He is our president. I'm waving, he's waving at you. You just can't see him. And we have all these wonderful people. And thanks to Becca Taylor for getting everything together. She's our wonderful program chair. She was the one who contacted you, and for admitting all the people that came tonight. And thank you for everyone for coming, and share what you know with others who weren't able to come. And we will also have this program posted on the Vimeo channel. And if you have anybody that you think, people in academia, people who are birders, you think they would be interested in it, well, we're going to share it in the video. So we'll keep it going. We'll keep up with you, and we will say, keep in touch.
For sure.
And we look forward to seeing that young man that you're
Yeah.
raising to be a naturalist as he grows as well. So thank you very much to everyone, and enjoy the birds and enjoy the rain that we're having when we're and we'll see you at some other event. We're going to be announcing some more outings and so forth soon. And watch our website and watch us. Come and join us. We'd love to have you. So we'll say good night and thanks again. Bye.
Thank you. Good night.